focus of AtlantECO is on the open ocean and also on coastal waters. But we have several collaborations with external partners and also with other projects to cover other environments or other aspects where microbiomes and plastics are involved. That includes aerosols, aquaculture systems, mesopelagic resources, benthic systems, deep sea systems, offshore resources, how they interact with microbiomes, and then land and rivers, and ultimately with humans as well. The objectives, also our activity streams in AtlantECO, we have four. The first one is to assess and map the status of the Atlantic ecosystems, looking at the structures, functions, the health and services that it provides. The second one is to enhance knowledge and innovate by enhancing observations, having new sensors, having innovative ways to do bioinformatics, and also developing new models. The third one is to assess change and to forecast the impact of drivers and stressors on the ecosystem. And the fourth one is to share capacity and transfer knowledge. Now, this webinar touches mainly on activity stream number 2 and 4. We want to share capacity and augment our capacity to make observations, to measure the ocean microbiome. Now, we do that with a number of activities. Some of them I will present. A first group of activities happen on flagship cruises with oceanographic vessels. So we have, for example, the Atlantic Meridional Transect going from the north to the south. That's implemented by the Discovery and the James Cook at PML. We have the Alpha Crucis in Brazil, working on the coast of Brazil. We have the Agulhas 2 from South Africa, sampling the South Atlantic, but also the Southern Ocean. Then we have a second class of flagships with research sailboats, notably Tara, will start very soon, end of July. We'll start to sample from the north of Brazil, going all the way south into the Southern Ocean, and then crossing to Cape Town and coming back up the coast of Africa. We have the Eugen Seibold from the Max Planck, who will sample mainly in the North Atlantic, and the Valerio Eco from Brazil, who will sample along the coast of South America. You can see that we will sample coastal waters mainly, also open ocean, but the R1 to 10, these are rivers. So we will go into estuaries and rivers to look at the link between their microbiome and that of the ocean, and also look at microplastic sources of microplastic and how they distribute in the ocean afterwards. Then we have activities with marine stations, and that's why we invited many of you from marine stations from the Ocean Sampling Day. So we want to work with the network of OSD, which you probably know. Most of the sites of the Ocean Sampling Day are in the northern hemisphere. There are only a few, maybe one in Brazil, a few in Africa. So we want to augment that capacity to do the OSD in the All Atlantic. So in 22-24, we want to have the All Atlantic Ocean Sampling Day. So that's an important milestone for us. Yes. I was asked to mention that this year, the Ocean Sampling Day on the 21st of June, 2021, in 1 month, this OSD will happen, but it won't be part of AtlantECO. It won't be the All Atlantic. It won't be the protocols that we are proposing today. It will be the standard protocols that have been used by OSD since 2015. So you've probably already been contacted about that. Then we have activities with citizens. So it's we call it the Citizen Sail for Science activity. So deploying a reduced set of protocols on sailing boats so that citizens can contribute. So they are quite robust protocols. Even though they are reduced, they can generate very good data. So that's one activity. Then we also have activities with moored sediment traps from north to south. And we will start the first CPR line in the South Atlantic between Cape Town and Rio. And what we also have is a hub and nodes approach, where let's start with the nodes, where we have a node in South Africa, one in Brazil, and one in Europe. And their role is to take care of metadata curation, biobanking, and sequencing and analyzing the samples for imaging. So the idea is that samples collected, for example, along the coast of South America by marine stations there would be sequenced, biobanked as much as possible in Brazil, and the same for Africa. And so the hub is composed of AtlantECO partners, and the role of the hub is to harmonize methods, ensure quality, and develop standard procedures with the nodes. And so that's part of AtlantECO. We also want to discuss with NOAA and with DFO in Canada to have a similar approach with the US and with Canada. So moving on to the system of standard protocols. Our system is inspired by Data Oceans. That's not surprising. That means that we want to deploy a set of imaging protocols and sequencing protocols that go across the full size spectrum of the ocean microbiome. And by that, we consider that it includes viruses, bacteria, archaea, protists, and also the small size multicellular organisms that together form what we call the ocean microbiome in the Atlantic. The system is also very much inspired by the international system of units. We aim high. So that system, everybody knows it. It's brilliant because it's quite simple. It's based on, it has 7 base units. They are well described. They have a clear semantic with symbols. It's a simple system. And it also includes derived units. And those derived units are sometimes simply a combination of the base units, and sometimes not so much. But what the system does, it provides equivalence to the base units. That's the philosophy that we want to have for our system of protocols. So we came up with this system of standard protocols for marine microbiomes. It has 6 base protocols, and they are partitioned according to first cellular organization of biological entities. So we have viruses, which do not have, they're not a cell, they do not have a nucleus, and they have either DNA or RNA. Then we have prokaryotes, which have a cell, no nucleus, but they have both DNA and RNA. And then we have the eukaryotes, which have a cell, or they live in colonies, or they are multicellular. They have a nucleus, and they have both DNA and RNA. The partition also takes into account the size of biological entities. So we have these generic size thresholds. So 0.02, 0.2, 2, 20, 200, 2000 microns. And now we realize that the complexity of life forms cannot be resolved and explained with simple partitions. That's clear. And we know that organisms will go across the bases that we define. And that's what the outer layers try to represent. So we have viruses, prokaryotes, and small eukaryotes that can happen, that you find in several of the bases of the system, either through different associations. It could be parasitism, symbiosis, it can be aggregation, it could be simply a bias of the method as well. But we recognize that they can be in different groups, and the system will need to address that. So that's. Then we have the 7th base protocol, the environment, which is conveniently put there to deal with everything that doesn't fit in the 6 first base protocols. So anything else could fit. Well, not everything, but it's convenient. For example, eDNA will be part of this component because it can target fish, birds, mammals, reptiles, things that fall outside the realm of our microbiome, but something that we use. We use that protocol in conjunction with the other standard protocols. So we want to address it in the system. An example of imaging protocol that would fit in that category is remote sensing, for example. So if we look, this is a table of the base sequencing protocols that we propose. So you'll see the 7 partitions. The label of the base protocols, we want to keep it simple, 1 letter, S for sequencing, I for imaging, and then a number of digits that describe the lower threshold of the size fraction. Then each base protocol is described by its targets. So targets in terms of entity, size fraction, the type of analysis we want to do with that, the volume, the time, that's about the processing of the sample, the target, the preservation of the sample, and there will be more specific characteristics of the protocol. We will, for example, specify or recommend what type of filter to use, what type of porosity to use, what type of tube to use to store the samples, what type of identification label to use. So there's a lot more that will come into the protocols, but these are the basic ones. I was mentioning we will recommend a pore size or a type of filter, but we know that, for example, to separate the prokaryotes and the eukaryotes, some will use a membrane with a porosity of 3 microns. Others might use a net with a mesh size of 5 microns. These specificities will be part of the metadata of the protocols that when you report your metadata, when you archive the data. But independently, these will form what we will call derived protocols, but they will all refer to the same base label. So whether you use 3 microns or 5 microns to separate these two, the base protocol label will be always SO2 and S2, and that's a mean to bring together data that are comparable. And then you need to go into the detailed metadata to figure out if what membrane was used and what size fraction exactly was used. But having standard labels will help in the integration with the data. Today, we want to talk about a package of protocols, of base protocols, a package of these 3, looking at viruses, prokaryotes, and small eukaryotes. The package can be achieved with 1 setup. So filtering on 3 micron, for example, 0.2, and then collecting the filtrate, flocculating it, and collecting it on a membrane for the less than 0.2. So that's one of the packages that we would like to deploy, and so we would like to develop capacity at the marine stations along the Atlantic Ocean to carry out that protocol. Similarly, if we look at imaging protocols, I won't say everything about this. It's the same structure. The target analyses are flow cytometry, fluorescence microscopy, flow imaging microscopy, flatbed scanning imaging, light microscopy, and as I mentioned, remote sensing could be one for environment. Today, we will focus on flow imaging microscopy as a key protocol that we would like to deploy across all marine stations. And that's what Anna will present later on. So that was the system it's the rapid overview of the system of standard protocols. What we need, I mean, we wish to deploy it, but it needs to be adopted by the marine stations. And for that, we want to build capacity. The first mean is to work with the Ocean Best Practices system of UNESCO. It will help to design the system, to further design the system, and it will mainly help to adopt it across the, well, Pierre will show what it is, but it provides mechanisms for people to learn about the system and to adopt it. Similarly, we will engage with EMBRC and MBON and Goose for the adoption of the system and also further defining it. We will have training activities on the different protocols, and we want to roll out sampling kits. Also, we have a special issue in Frontiers that's managed. It's a joint special issue by AtlantECO and the Aura Marine Microbiome Working Group. And so that's one place where we hope to get the community to publish about their best practices. And eventually, when we wrap up everything, ultimately, that's where we will publish the system of standard protocols as well. And with that, I think, yes, it's a good time to pass on the screen to Pierre. Stop sharing. Pierluigi, are you there?
I am. Can you see my screen?
Yes.
Great. Okay. So thank you, Stefan. It's a great way. So thank you, everybody, for coming. And I'll provide some perspectives on what we've been doing on the IOC, IODE, UNESCO side to help gather up all of this methodological knowledge that projects are generating, because a lot of it, unfortunately, is lost when the projects are done. There are some papers that are published, but a lot of the actual base methodology, really well written that you can follow in a lab, and standard specifications and guidelines and also policies that they care about, that sort of evaporates over time. And then you'll have to find somebody, hunt them down to get that information. And we'd like to change that. So I'm wearing several hats on AtlantECO side. I'm representing the Alfred Wegener Institute. I work for the Helmholtz Metadata Collaboration, the Earth and Environment branch, but I also contribute to the OBPS on behalf of IODE of UNESCO. So let's jump into it. The reason that we're working with this fits very nicely with the AtlantECO goals. We're trying to build that capacity, as Stefana mentioned, around taking in the protocols that are standardized by something, in this case, a project, right? We're not a standards body, but we are standardizing enough to make coherent observation possible. And then eventually make them into best practices. And again, best is qualified, best for this project, best in the given circumstances that we were operating in. And those circumstances are as important to communicate as the methods. And exactly, once you have that, you can start generating higher quality, more coherent, globally aligned observations. And build better sensors, of course. Right, the problem that the reason why we started putting so much effort, and this is a lot of people, a lot of voluntary time, a lot of people believing that this needs to be done into the ocean best practice system, is because most of our ocean practices are scattered around. They're edited in all sorts of formats. They're usually sort of squeezed together into a PDF and put online for somebody to view on a website. This part looks great for us. It's fancy and all of that. But as soon as you compress all of that into a PDF document, you've created what an artificial intelligence or a machine agent will perceive as a horrible nightmare. It's almost impossible for us to then text mine those and get things out automatically. And there are thousands and thousands of these being generated. So we're losing a lot, not because they're not accessible, but just because they're not accessible to machine agents that'll let us go over the hundreds of thousands of documents that are produced probably every few years by European projects alone. So that's the idea. And also the fact is, when you come back from the field, back into the lab, and you want to look for those protocols and best practices for a certain project, some of them you will find. Some of them, the project website is still live. Sometimes the project website is long gone. Some of them have been properly archived in the European system, but some of them are coming from other countries and nations who may not have the same sort of setup. So again, there's a big leak in that system, and that's the risk. And that's why, again, our mission is to create a fairer future for best practices, but any methodological content generated by the millions of euros that the public put into our enterprises, again, every year. So it's borrowing the FAIR concepts from the data side, but we have the same thing with these methodological artifacts. They need to be findable, accessible via the web, interoperable, so that if you find one, you know how it relates to another, and the system storing them can talk to each other somehow to say, hey, look at what I have. Do you have similar protocols? So that we can all reuse them and actually align and advance together, as opposed to reinventing things, which is quite often done again. So the Ocean Best Practice System was set up. We've been working on it for a number of years. Again, lots of people, lots of voluntary time, a lot of passion going into it. And there's our website. You can read a little bit more about our mission and our setup. And it's moving along through tireless efforts from many. It's now part of many projects, and we're attracting methodological archiving from a large number of organizations, not just in academia, further afield too. And that's encouraging, because people also see the same problem. They have this stuff on their institutional archives, but they'd like to see what other people have, and they'd like it to be secure for the future, particularly when the UN decade starts of ocean science, very soon, in fact. So the general setup of the system is that we have a number of participating organizations. There you see some of them, and they will be contributing documents, methodological documents, standard specifications, guidelines, policies to the central repository. That's a long-term repository. It issues DOIs and all of that nice stuff. So you get your metadata locked into to the documents themselves. And it's not just documents. They could be videos, they could be audio recordings, they could be any kind of multimedia. We also have partnered with a ongoing research topic in Frontiers. So in case those stakeholders need to get academic credit, they can still publish their method for academic crediting, and we can cross-archive it in the repository. Similarly, we can cross-archive any journal article that has the appropriate licensing. Once it's in there, we have a stack of technology, our enhanced discovery services, that tag up that and plug it into data systems. I'll talk a little bit more about that. And then we deliver a front-end experience to users to help then query across that entire corpus of methodological material. That's a basic setup. So I also wear a hat in the Goose BioEco panel, and I think this is also particularly relevant to AtlantECO and related projects. There are these essential ocean variables for biology and ecosystems that focus on different compartments or different functional groups in the ocean. We've been partnering with Goose and the OBPS to have an endorsement system where panels like the Biology and Ecosystems panel can tag methods in the system to say, yes, we recognize this as a valid way to measure this essential ocean variable. We've drafted the technology. Gooses have a workflow for this to happen. And so we're very excited about that, because this will help people find things that have been curated and have been quality controlled sufficiently to say, yes, this can actually feed into a global system of measurement. We're concerned about that in some ways, because many projects want to contribute to the EOVs, but on the Goose side, we need to help them, guide them to how to do that most effectively. This endorsement system and endorsement technology is not limited to Goose. We want to generalize that. So as we saw here, it's entirely possible that we can support things like a Marine Station or EMBRC or any other group endorsement process to say, yes, we as a consortium of professionals think that these methods work very well for our aims, capacity ranges, etc. And that's really, really important to help people find them and understand why they are the best practice in a certain group. So that's something to think about. But again, once that document or multimedia thing has been submitted into the Ocean Best Practices repository, it's safe, it has rich metadata, Dublin Core, it's interoperable. We also link to UN data systems for the Sustainable Development Goals and other systems through that metadata entry process. So it takes about 15 to 20 minutes. Once you've done it a few times, it takes shorter to upload these documents. And we are working on automated metadata extraction too for the future releases. But once it goes in there, then exciting things start happening. You upload your best practice into the system. And once it's there and safe, we have a stack of technology that uses ontology, so knowledge representation technology, artificial intelligence agents, and standardized vocabularies and thesauri to tag up that document. So we rip the text out of it as best as we can, because most of them are PDFs. And as I said, that's a machine nightmare. But you can usually recover some text, and we tag it up with those ontologies. So then that machines understand what's inside those best practices too. They sort of read them and they understand the links to them. And then we can present them into the search interface with technology that's ready for a future of more advanced digital technology operating across multiple systems in the ocean datascape, as it were. Up here, that's the interesting part, because we're reusing a lot of those ontologies and vocabularies already used inside UN systems and broader systems in the regions. So again, special interest to AtlantECO. They have just been extended through the work of Raisa Meyer and her teams, also AtlantECO affiliated from AVI, to include the marine plastics, which UN Environment is likely to adopt. We have a meeting with them upcoming where we discuss what kind of ontologies can support their needs for the official reporting of the SDGs. So we're bringing research closer to operational reporting, and that's the whole goal. All right, so that's what it feels like. It looks really simple. There's lots of this technology in the back end, but we try to keep it simple on the front end. You put in your search term, you'll get some results back. Looks pretty simple. And you can check out the tags. You can check out the semantic tags that I just explained. And it looks fairly simple. It says, okay, an ontology has found the following terms in there. And if you click on them, you do a little knowledge neighborhood exploration. There's a lot more you can do, but at the moment, this is where we're going to push forward. And you can add them dynamically to the search. So it enriches the search interface right off the bat. But more importantly, in the long term, this is another hat, for the sculpting of the ocean data and information system on behalf of UNESCO for the decade and beyond, those same semantic technologies, that's the I in FAIR, that's what's going to help us link the OBPS up here into a global linked data universe that we're sculpting across the various partners in the UN and also outside of the UN, in Europe, but certainly beyond. Our Ocean Info Hub project, you can check it out online, is building those interoperability layers between the regions, so Latin America, the Pacific Islands, Europe, beyond. So it's all happening, it's all very exciting. And all it takes at the first stage is to just submit documents and fill out some metadata to plug into this system. And all of this magic stuff happens in the background and helps make sure that these things don't disappear after the project ends. So 10 years later, they'll still be plugged in. But of course, we can improve it. And how do we do that? We published some guidance as an IOC manual and guide on how to write your protocols in a way that would best work with the systems we have right now. It's a compromise between being able to work with it on your PC and also then submit it to a machine readable system. So it's worth checking that out. I'll drop a link to that and a associated perspective article in the chat when we're done. But this can already help greatly. We've thought about that in the whole process of how those practices are developed and how they're refined. And again, the Ocean Best Practice System tries to support each one of those steps, including version control. So as a method is updated, you can update it in the system and still log the versions as it goes along. And that's very, very important to show how things have progressed and how things eventually converge. So multiple practices converge into one. And that's what we love to see, because that's when people, expert groups find each other and come up with unified solutions. So inside that document, again, we just lay it out into how you can separate your practice out, what parts can be read by our machines and what parts cannot be. These include tables like our metadata tables, and I cheekily, before we get there. And other projects are also adopting this. So Tech Oceans, it's running right now, started in October. This is developing lots of sensor technology, including sensor technology for eDNA and omics. They are following this step, creating their own space in the OBPS. and creating templates for each of the major types of protocols and standard specifications, etc., that they need. So we would very much encourage stakeholders to do the same. It's very easy. It's a Word document. Yes, we just try to keep it as simple as possible. And once you do that, you're already future-proofing a lot of your methods. So that's something we're very excited to do. And again, that has bearing on this system of manuals that Stefane has been talking about. I took a screenshot of his presentation because these tables, if indeed we can standardize to that degree, we can also make all of this machine readable inside the OBPS. And that's where things can get very exciting in terms of search and discovery. We're moving towards this world. There are some things in biomedicine, for example, that are already using this, highly structured methodological documents where you can have natural language queries and then return things that make sense. So gradually we work towards that and then make sure they're all connected to marine data. Yeah, so all of this fits in wonderfully with the standards and best practices. And we hope that we can make this link happen. And I think we're very confident that we will. We will create a space inside the OBPS for the work associated with AtlantECO. And so there will be the presence of the project, but also the backend technology makes sure it's not isolated and linked elsewhere. A workshop last year created a working group on omics and eDNA. Raisa Meyer from ABI was the AtlantECO link to that. And that was a very interesting bridge to it. They helped us in the OBPS strategize of how to support the broader omics and eDNA community, and we're following up their advice, so much so that this year, in the upcoming workshop in September, that working group has transitioned into a task team inside the UNESCO OBPS, and would very likely, we have to send out the call for contributions, but would very likely have another session. So perhaps that's another chance for us to bring together AtlantECO and other stakeholders who are interested in developing these methods together during that week to figure out how to do it. We're also coordinating with GeoBond through a proposed omics bond. Hopefully this is in consideration, and a submitted Ocean Decade program proposal for a biomolecular ocean observing network. So we're, again, converging all these stakeholders over shared systems. So to close, we can leverage a lot of technology. We're doing a lot in the background, and we're trying to keep the front-end interaction pretty simple. A Word document, standard template. Once we can do that, we can start syncing across not just the EU projects, but also international projects, and we encourage you to be part of that. And we're linking to the large, as many communities as we can, especially communities that are trying to align practice. That will help us develop things iteratively, transparently, and also in a machine-readable way, getting ready for that future. And the main thing that gets to me is that this helps us publish methods faster than we publish papers. And I think that's very, very much needed, because too late we say, oh, wait, my paper will be published at the end of the year. And then already somebody's invested a lot of money developing something that you could have helped with if you published the method earlier. It's a change of a sort of academic norm, but it's much needed. Exactly. So I'll close with that. And I think that's an overview. I think I went on long enough. Thank you, Stefan.
Thank you very much, Pierre. Very nice. And let's look into having maybe converging or joining the workshops that we're planning. Let's see if we get fatigue out of all the workshops and webinars.
Yeah, I can sympathize with the fatigue. I can't.
Yes. So next we have Anna to present the planktoscope and an imaging kit.
Yes, I think, hello everyone. So until the very last minute, it was not very clear whether Colomban and Fabien could make it to the meeting. So Colomban is actually here. I don't know whether he wanted, I think, to introduce some slides, Colomban.
It's really up to Stefan. I asked Stefan if you want, because Anna will speak mainly about the imaging system we have. I wanted maybe to say a few words about what we did for the genetics above 20 microns, but it's really up to you, Stefan. I can do it in 2 minutes.
It's just that we because of technical glitches, we had to start 10 minutes late.
Yeah.
So if you want to say a few words, yes.
Yeah, just a very few words. So I will share my screen really.
Yes.
Just partager écran.
So I introduce you a special guest, a surprise guest.
Can I share my screen? Yeah, very quickly.
Thanks.
So just in the frame of this program Plankton Planet, our belief is that we need to develop frugal tools, meaning that those tools are cost effective, scientifically robust, built by communities outside the academics. So also with maker labs, etc. So we are building together with Manu Prakash at Stanford University and a group of engineers that is growing now, a new generation of instruments at the size of the complex at the spatio-temporal to fit the spatio-temporal complexity of ecosystems. We think that instruments will be the key for understanding global patterns of ecology, and mainly in the ocean. And so what we have done in this context is that we build up a very simple protocol to collect plankton larger than 20 microns with this plankton DNA kit back in 2015. And so this is really plankton larger than 20 microns. And we're asking people on sailing boats to dry this plankton, to send it back just from regular mail. So very low technology. And we send this protocol in I think 20 boats at this time who sail the world ocean. And in 1 year, we got the first citizen plankton data sets that we compared. So we sequenced barcode out of this data set. We compared it to the Tara Ocean data, and it was about this. It was the same quality in terms of of what we got. It was perfectly making sense. We could detect very interesting patterns, and yeah. So this is plankton really larger than 20 microns. This is published in bioRxiv. We need to push it to a new.
Thank you, Colomba.
And then just one thing. So we are developing also instruments to collect plankton at high speed, and the planktoscope. And what I wanted to say is that we are really targeting plankton above 20 microns with these things. Yeah.
Good. Thank you. And that fits with Anna's presentation.
Okay. Share the screen. Okay, can you see my screen?
Yes, but not in full screen mode.
Okay, it's coming. Slow computer. All right. So I will try to be as little redundant as possible with Colomban, who actually introduced you the big picture of what we are doing. So I'm the project manager actually of Plankton Planet, together with Clémentine Moulin, that many of you might know. And the Plankton Planet is really at the heart of this subtask of AtlantECO. So what we are interested in is quantitative imaging. And this is a type of imaging that allows for extraction of reliable numbers from images. So quantitative imaging implies a number of criteria. First of all, images must be taken in a highly standardized way. The whole sample must be imaged. We try to measure a maximum of morphological variables, and we collect the metadata associated with the images, like geographical coordinates, time, of course, volume, sample, fraction. And these metadata are just as important as the images themselves. So in practice, how does it work? What we do is we run a sample through an imaging device and we take pictures. We are not looking for specific components. So we try to image we do image at random position at regular times. And the images that we get are then segmented in order to get some region of interest. And each particle that is segmented gives a vignette. We get thousands of vignettes. And on those, we can measure a number of parameters like size, length, color, and so on. And via both artificial intelligence and human annotation, we can gather a lot of information out of these images. There are, of course, advantages and limitations to this approach. So the main advantages are that this is a quantitative way of looking at images. So we can retrieve numbers, such as in situ concentration, size, and so on. It is collaborative, meaning that we can compare samples across different stations and different laboratories, as long as they have been acquired and analyzed in the same way. And it is easy to record the results in a digital format because we have both images and numbers as results. Also, we can explore features at the individual level. So we have the whole sample where we can zoom in individual particles and look at the differences in each of those. The main limitation is that we can rarely identify down to the species level. So we often stop at the order of at the level of the order or the family level. So we have kind of a low taxonomic resolution. An important point to bear in mind is that each imaging technique, plankton imaging technique, is adapted to different ranges of plankton size. So plankton, of course, spans across several orders of magnitudes. And here in red, on this table, you have an example of the imaging techniques that were used on Tara. And in red, the effective size ranges that can be used for quantitative imaging, and in dashed instead the commercial information ranges. So during the Tara Ocean expedition, we gathered something like 8 million images, going from 0.1 micron to 10 centimeter size. And as you can imagine, 8 million images is a lot of images. So in order to be able to organize and analyze all the data generated, in 2016, EcoTaxa was created. So EcoTaxa is a platform. It's an open access platform where all the images generated can be deposited. We now have around 150 million images, and actually 4 million new images every month are deposited in EcoTaxa. There are more than 1,000 users registered, and roughly 50 to 60 active users every day, meaning that it's not just a place where people deposit things, but it's also a place where people retrieve information. It is based on both machine learning in order to recognize what is in the images, and human annotations. So you have a first step of artificial intelligence followed by human validation. This is still ongoing work. A lot of development are still needed. There is a manuscript, I believe, in preparation on this, but we believe this kind of database will be crucial for being able to create a large picture of the ocean. Now, we are interested in citizen science, ocean citizen science. Colomban has already introduced to you the DNA kit. What we want to do is basically to take advantage of all the people that are sailing around the world in order to increase with a very low price the spatial and the temporal resolution of ocean sampling. This was tested in 2015 with the DNA kit, a very simple DNA kit that, as Colomban explained, was just meant to collect plankton on a membrane with a manual pump. So there is no energy involved apart from manual energy, because we know that sailing vessels very often have energy limits. There were no toxic chemicals involved, also for the same reason. And it was really a very simple system. And what we got out of it was, first of all, that the quality of the data was very similar to the quality of the data acquired with Tara. But the other interesting thing was that the feedback from what we call the planktonauts, so the sailing people, that citizens that acquired the data. And we had mainly two types of feedback. The first was that they were not very happy because they had to slow down in order to collect plankton in a net. And the other feedback was a bit their frustration because they could not see what they were actually working on. They wanted to have, as a reward, the ability to see the plankton. So based on that, the following step was to try to develop a low-tech, frugal, but robust and reliable system for imaging plankton that could be used by non-scientists. And so Thibaut Paulina, in the framework of a collaboration between the lab of Colomban and Roscoff and Manu Prakash lab in Stanford, started developing what is called the plantoscope. So a very low-tech microscope that can take reliable enough images of plankton and that can be used on board of sailing vessels. So here on the left, you actually see how the plantoscope looks like. It's just a very simple case with a Raspberry Pi computer, a camera that goes with it, two very simple lenses, and a peristaltic pump for the microfluidic system. And this is then controlled by a software written in Python that controls all the different parts of the microscope and associate to images all the metadata that for the moment are still manually input in the system. The first thing was to compare the quality of the images that we're getting with respect to the commercial systems. And so what you see here is on the left, our plantoscope, and on the right, the FlowCam, which is a gold standard for plankton imaging. And below you see images in black and white coming from the FlowCam and in color coming from the plantoscope. And you can maybe already appreciate that the quality of the image is very similar. And in any case, what we saw was that indeed the resolution was very, very similar, and the images could allow for identification, taxonomic identification of the plankton down to very often to the species level. We also made sure that actually the distribution that we were getting of plankton out of the two instruments was similar. This is also published in a bioarchive, so you can look it up if you want. And in January, February this year, we did a second big test of the plantoscope on board of Tara during the trip that brought Tara from France down to Punta Arenas, where the microbiome expedition started. And this is also confirming to us that both systems see the same population of plankton with the same quality. We were actually testing as well a new net for collecting plankton. That's why you have, for each station, you have 4 data sets. So basically 2 samples were acquired collected either with the high-speed net that we've been developing. So a net that allows for collecting plankton above 4 knots and below 10 knots, cannot go faster than that. And the deck net that is normally used on board of Tara. And each of the 2 samples was then split in 2 and imaged either with the FlowCam or with the plantoscope. And so I'm not going into the details here, but the summary is that we are getting very promising results. The quality of the images is very similar, and the populations that are imaged with the plantoscope and with the FlowCam are very, very similar. Now, this is, of course, still work in progress. We are still testing the plantoscope further. We are developing new prototypes. And the main points now, the main challenges now are to make it user-friendly for a non-scientist, so more automated, to make it robust, because you need to resist on board of sailing vessels, and to make sure that it does give reliable results. And thank you for your attention.
Thank you very much, Anna. And my closing remark on that is that the plantoscope is much, much cheaper than a FlowCam, and it's much more affordable for marine stations along and across the Atlantic. And that's the plan, to, as much as possible, roll it out to the marine stations so that we can start imaging the same thing with the same instruments across all marine stations. So let me continue. The presentation. So we asked you, the participants, to fill a survey, and I want to thank you. We had 40 responses. That was a very quick survey, just brushing, having an idea of what are the best practices in the marine stations and what is the capacity in the marine stations for protocols on genomics. So a few slides on the results, and then I'll talk about the filtration kit. Oops. So we had 40 responses, people sampling from jetties, from shore, from the beach, from recreational boats, from small, medium, and large research vessels, and also commercial vessels, sampling with buckets, niskins, pumps on deck, in situ pumps, plankton nets. So a wide range of sampling capacities. So that's interesting. Then what we asked about some specific components of the protocols, we asked, what is your best practice? Do you have capacity? And do you wish to develop that capacity if you don't have it? The pink part, we have capacity. We have to interpret carefully, because some people will say, this is our best practices, we have capacity. Others said, we don't have, this is not our best practice, but we have the capacity if we want to do it. So the pink part can be interpreted in different ways. So the first question was about what type of membrane filters they were using. We see that best practice go across different, the full range of what we asked, GFF, Sterivex, or membranes. What we propose is the membranes as a standard protocol if we want to look at viruses, prokaryotes and eukaryotes, and do the ecology on that. There's also quite an interest on the Sterivex, I think mainly because of eDNA becoming more and more popular, and the Sterivex is, from the start, was the main method for eDNA. Then it becomes more interesting when we ask about the size of the filters and the type of holders that they use and the type of pumping that they do. We see that the most used best practice in the community is 47 mil cups using vacuum pumps. It's not necessarily a correlation between the two, but I know that it is. And these 47 cups most likely have a mix of membranes and GFF filters. And that's not surprising. I mean, for decades, it's been the standard for studying plankton. Well, let's say phytoplankton mainly. So that's not surprising. This is the best practice that is mostly spread in the community. Second comes 47 mil filters in Swinex, most likely with peristaltic pump of medium capacity. Again, most likely using membranes and GFFs. That system can also be used with the Sterivex. Sometimes people mix 47 mil Swinex with a Sterivex to do some size fractionation. So that's good. Then the least spread best practice is the one that we propose, and that's not so surprising. That's one using larger membranes, 142 mil. It requires slightly higher capacity peristaltic pump. That could be a mix of GFFs or membranes. But obviously that's a method that is more expensive to get the equipment. So it is less spread at the moment. But there is still some stations having it as best practice. And most interesting is that there is capacity and there is interest to develop that capacity. So that's a very encouraging message that we get. Similarly, if we look at, we asked how many size fractions do you use in your best practices? Most popular one is 1 fraction. That's coherent with the most popular. If you use a filtration cup or a Sterivex, you tend to go for 1 size fractions. That comes 2 fractions, and finally 3 fractions. That's our proposed standard. So 1 for viruses, 1 prokaryote, 1 eukaryotes. And again, there is lots of capacity, even though it's not the best practice. And there is interest for those who don't have the capacity, there's interest to develop it. So again, very promising. Same with how much volume do you filter? Not surprising, if you have a small filter or a Sterivex, you cannot pass through so much volume. So the best practice is less than 5 liters. Our proposed best practice is 20 liters. With Terra Oceans, we went up to 100 liters. That's not easy for marine stations to keep up with. But the 10 to 20 liters seems to be adopted by a lot of stations already. There's lots of capacity and lots of interest in developing that. Again, very encouraging. And the same shows up with when we ask about how much time, do you have a time limit to your filtrations? A max of up to 60 minutes, that's typical for Sterivex and small size membranes. We want to go to maximum 15 minutes. We'll look at the reason why in a second. But again, lots of capacity and lots of interest. So overall, I'm very pleased with the results. It was a bit of a gamble to ask these questions to the community, given the type of protocols that we want to push. But I'm happily surprised. Not surprised, but happy about the results. So what we propose as a standard, the ambition is to study viruses, prokaryotes, and eukaryotes. Nothing's new. And to analyze meta B, meta G, and meta T on them. The requirements, we need a relatively large volume, especially for the eukaryotes who are less abundant. In a short time, especially because we want to do meta transcriptomics. So the protocol that we aim for is 20 liters or 15 minutes, as shown already. And that's also doable in coastal stations. If you can only pass 7 liters in 5 minutes, fine, it's documented, and it meets the target of the base protocol. It's 20 or 15 minutes. Another requirement is size fractionation. That's mainly, we want to have on two different, one filter dominated by eukaryotes and one filter dominated by prokaryotes, so that when we go to do sequencing, the very abundant sequences of the prokaryotes are not shading those of the eukaryotes. Having them on separate filters, sequenced separately, you can go much deeper in discovering the eukaryotes. So obstacles. Obviously, as we said already, it's more expensive, and we need to address that in AtlantECO and beyond and find solutions. And it can also be logistically challenging in some situations to collect 20 liters. If you want to have replicates, 60, you go up to 60, 100 liters that you collect. That's a water budget that is sometimes difficult to reach. Also a setup with 142 mil filter holders and a big peristaltic pump that can take some space on a bench. So these are challenges that we need to address to improve the uptake of the proposed standard. So what it looks in terms of materials and methods, pushing for a Masterflex IP peristaltic pump. We name the brand. We're not scared. A millipore, 142 mil stainless steel filtration head. And what you see on this side of the screen is what we develop as a plug and play filtration box. It's developed by Sorbonne University and Ternog Lab in Brittany. And the idea for this is not just to have a fancy looking box. That's not the objective. When you do filtrations with these big filtration head, many little things can go wrong. And marine scientists, we're very good at troubleshooting and solving issues in the field. The problem is we don't always solve them the same way. And that's an obstacle to having reproducible methods. Also for the quality of the sample, it becomes a lot more variable depending on the people who do the protocol. So the idea with the box is to have something that is user-friendly, it's clean, it's you can focus on, it might look a bit strange to say that, but you don't focus on all the things around. You focus on your filtration heads and your filters, because what can go wrong is usually the step when you set up your filters, when you remove your filters at the end of the filtration, draining the membranes. The idea of the box is to simplify all that and to reduce the risks. And if something happens, to reduce to force the user to solve the issue in a certain way, rather than being inventive and doing lots of different things. So the box assembles 2 filtration heads solidly on the box. So it uses standard instruments. They can be used in parallel. They can be used in series to do size fractionation, as we want to do. Basically, it removes all the clutter of the 6 legs and the tubing that goes on the bench. So again, as I said, you can focus on the important parts, your filtration heads. Underneath, we have elbow connectors, so it reduces the overall height of the system. It makes it more stable. On the top, you don't see that on the image, but we'll have quick release connectors, which makes it even more plug and play, and will make it easier to drain the filters, which is a crucial step at the end of the filtration. You have slots on the side to keep everything tidy and clean. You can just put your the top part of the head. So it's making life easier in the field with that sort of setup. Yeah, so the full cost. The box doesn't cost so much. It's really the big parts, and we need to address that. And it's the same with the imaging system that was presented before. The difference of price between a flow cam and a plantoscope, I think that the plantoscope now is between 500 and 1,000 euros ish. 2,000? 2,000 now. It's going up. But again, so it's much less expensive than a flow cam, but it's still 2,000, and we need to deal with that and find solutions to make it affordable for the stations. So the target is to start using these kits progressively during the AtlantECO Ocean Sampling Day, starting next year in 1 year, in 22, and then 23 and 24. That's for within the scope of AtlantECO. Then yes, I should finish soon. Then hopefully we can continue with that network beyond AtlantECO. And we want to start using the kits on cruises in the Atlantic. So Tara will start Mission Microbiomes for AtlantECO in July this year. It will have 5 units like that on board to trial it and improve it. It's also being tested in a marine station in Roscoff, in Brittany. And so the next steps also involves having sponsorship. So we want to contact UNESCO and the capacity building offices of UNESCO in Africa and South America, getting their endorsement as part of the UN Decade for Ocean Science. That would be a big plus. To get sponsorship, we will get in contact with Masterflex and Midipar to get discounts. And we want to work with you, the marine stations, to work on co-funding opportunities. If you could work up ways to fund a kit, having a kit rather than just bits and pieces, having a kit that is used broadly in a network, I think that's much easier to sell as a research investment. So it could be co-funding at the institutional level, across institutions, as a network, nationally. We have to see what is possible. But so AtlantECO has some funds to equip the flagships of AtlantECO with boxes, but for the marine stations, we can only help with sponsorship or, well, support. Then we're planning training in South America and Africa. So on board Tara and ECO, when we do port calls, we will invite marine stations on board to get trained on the protocols in general and the kit. Training in Europe will happen with the help of EMBRC. We'll have a spring school in 2022, also preparing for the AtlantECO OSD. And then we have to think about ways to roll out the kits to the different locations. And that's it. I think now, yeah, well, we're not too bad. We don't have so much time for discussion, but let's move already on share. Eloise, do you want to explain how you want to work with the questions? And then we can start.
Sure. So we had a few questions already in the chat. There's been a couple of discussions. I don't know if they've all been resolved or not, but I can actually share.
And what I would like to discuss or to hear from the participants is how interested are you to adopt such kits, so a planktoscope and a filtration kit, moving to larger size fractions and a larger filtration head. Oh, wow, that's organized, Eloise. Thank you.
So I tried to organize it by conversation because it kind of got mixed a little bit. So there was a first question for Colomban from Eric. Let me see if I can just allow you all to unmute yourself now.
Yeah, so Eric, do you want to?
Actually, Stefan, you should be able to, because I'm sharing my screen, you should be able to unmute everybody or allow them to unmute themselves. You can't? Maybe I'm the only one that can do that.
Yep, seems so.
All right, people can unmute themselves now.
Okay, this is Eric Zettler. Can you hear me?
Yeah.
Yes.
Yeah, we've had this issue quite a bit. I work with Linda Amaral Zettler and with citizen science, which is a great opportunity, but with the regulations for research clearance, most citizen scientists aren't able to really do that on their own. It's hard to do it up front, and it's even more difficult to do after the fact. And really the fact is, most countries would interpret that if you are taking any kind of data, doesn't matter whether you put the sample back or not, if you're collecting data in their waters, technically you should have research clearance. So that could be something that as a community we have to think about in order to take advantage of these citizen science opportunities.
Yes. And one solution is to sail in international waters.
If I can say something, I think it's very difficult. You know, for instance, I was speaking to Jan Salter or someone in Denmark, and he had absolutely no problem with that. And then if you speak about images, nobody knows really. So I think for DNA and if you take biomass, I think there may be some regulation, but you know, I don't think it's that clear for me.
No, but even for underway sampling for sensors, it's also an issue. So that needs to be addressed quite carefully, especially for sailing boats. Yes.
You think if you take an image of plankton,
it's data.
If you take a plankton sample and you take an image and you.
But I would like us to focus today on this webinar on marine stations and oceanographic vessels rather than citizen science, because that's the audience that we have.
Yeah.
Frank has his hand up.
I know. Is this in response to the question on research clearance or not? Or is this an additional question? Okay.
Well, you asked if we can address the comments that we put in the chat. I can wait.
It's okay. I was just going to go down the list as they were asked, just to see. I think your question is the one after this one. There was a question from Pierluigi. I don't know if that was answered or if you want to discuss that a little bit more. Are your annotations using well-adopted controlled vocabularies, ontologies, or taxonomies that provide web-resolvable IDs for the taxa?
Yeah, so that's a concern that we also have in other projects. There are people doing labeling of images with AI, but those labels are just strings. They don't connect to anything, so nobody else understands what their system is doing. And there are internal standards, which is good. It's good to get your house in order, but they don't talk to each other, these systems. So it would be very interesting for these projects to start talking to each other so that all of these different AIs that are annotating images can at least share with each other what they have found in a way that the machines understand. So that's all. I think this is fairly resolved. It's just something that I'm also urging the TechOcean side to do too. Ecotaxa is involved there, I believe, also. So yes, I would really urge whoever's working on that to try to build some capacity in that kind of ontology, web vocabulary, and of high quality too, because that feeds into all of the data systems that we can connect to the best practice documents and to OBIS records and to INSDC records. It's the back end. So please do think about that.
Okay. Great, thank you. So Frank, maybe you want to follow up?
So one of my points was related to this. I agree with Pierluigi, and Colomban also replied. So it is important to have the vocabularies, but also the data formats be interoperable so that we can migrate these data into common databases. And at least 3 were mentioned in the chat. So this is really critical as we move into the ocean decade and how we can integrate and have these observing systems and projects collaborate. So ultimately, we would like to go to some simple search mechanism and find that the biodiversity data that we need, or the abundance data that we need, in some relatively common format with common tools. So that's just kind of the follow-up to what Pierluigi just mentioned. And I know that there's a lot of work being done with that, but it's never, I guess it's never superfluous to restate that and make sure that this program also serves as a model for how that can be done.
Maybe to answer your question, Frank, so OBIS, UNESCO OBIS is partner of AtlantECO, and they're the main, when it comes to that type of data, that's the end of the line. That's where all the data will get.
Great, wonderful. And my other question was, how do so I represent one of the Marine Biodiversity Observation Network projects in the US. There are 6 projects. We all sample plankton in one way or another, and there's a backlog in how we analyze those samples. So this modular plankton camera sounds very interesting. And I'd like to be in contact with you and see if there's a way to test that over here.
Excellent. Yep.
Thank you.
From Colomban, Anna, do you want to respond, or maybe just get in touch offline? Yeah.
Yes. Just a short answer. We will have a system that is stable enough, hopefully during this year, and we will contact you directly, and there will be a website, where people. Yeah, the idea is to make it available to a maximum of people, and in fact, we will also release a plan of the system. So yeah.
It may be good to know for everybody, Stefan. There's a SCORE working group 154, that made recommendations on how to sample plankton and the groups that you're looking at in an operational sense. Okay, so you're probably familiar with that.
That's with Anya Wade and Emmanuel.
Yeah, exactly. Right. And so they are now moving to an implementation phase with GoShip that was funded by NOAA as a pilot for 2 years. And it would be really good for you, for example, the filtration kit and some of the protocols that you're using to crosswalk that. If GoShip actually adopt this after the pilot, it would be really good. And if you have knowledge or experience or suggestions, it would be very important to crosswalk that with that group.
Yes, definitely. That's in the plan.
Yeah.
I mean, we already had contact with GoShip in the planning phase of the project. They are in the plan. and we've been in discussion late last year, not so much recently, so we have to get back to that.
Yeah, but the funding came recently, and so it is happening now, and they plan to get in the water in early 2022. So it's a good time to talk.
Excellent, thanks.
Great. So I'll move on to the written questions before we can take the if you want to raise your hand later on. So there are two questions for Stefan. The first one is, will the filtration kits be available to borrow for specific cruises of opportunity? And this is from Eric.
That's the goal. So we will have a few, we want to, so there is some funding in Brazil, South Africa, and Europe. So we could get, let's say at least one kit in each region available to be deployed on a temporary basis on board cruises. We already start discussing, so there's a cruise, Amazomix, some of you might know. We're trying to get a kit on board, but at the moment we have to look at partners who might have the pieces, then we bring them together. So at the moment we're slowly getting there, but that's the aim. In 2022, having a few kits that we can deploy temporarily on cruises. That's the plan. Gavin. Austral, yes, the solstice. Yeah, that's a long discussion. Every time we discuss, we reach a different conclusion. We have to get back to that discussion. The last time we discussed it, we said that it would be 21st of June in both hemispheres, but we're open to discussion. We could, so for the All Atlantic, we will start with the 21st of June, 2022. And then let's see, we could do 21st of December, 2022 as well.
Okay, great. So now we can take sort of live questions. You can raise your hand, or if nobody's raising their hands, then unmute yourself and ask. Oh, Eileen has a question.
Hello, I'm Eileen Bresnan from Marine Scotland Science, based in Aberdeen. I just have a question. I might have missed it because I was called away for 10 minutes during your presentation. Once we filter the water on the filters, do we need to send them on dry ice to the analysis hubs, or are we going to use this citizen science protocol that Colin developed?
We wouldn't use the protocol that Colin showed, the drying of the filter.
Yeah.
Even for Sail for Science, okay. They might be moving to using a buffer. Mm-hmm. So yeah, that's a question. Do we ship on dry ice? In AtlantECO, during AtlantECO, we work with World Courier, so we can ensure the chain of cold.
Okay.
But beyond AtlantECO, that could become an issue. So we have to think about dry ice or maybe a buffer.
Yeah, because we had quite a few issues with the Ocean Sampling Day and getting it delivered on dry ice.
Yeah, I heard that.
Yeah. Okay, thanks.
Thank you.
Raisa has a question as well now.
Thanks, Eloise. I just thought I'd mention, because I'm involved in the OBPS task team that Pierre mentioned, which is working on developing a structured way for protocols for omics and eDNA, that we're also working on developing templates. And I think that might actually be a very valuable opportunity to synchronize with what is happening within AtlantECO, either in the review phase or earlier, to make sure that whatever is being developed on that side can also work for AtlantECO and vice versa. So if you'd be interested, please do reach out and we can make sure that we synchronize on that.
Yes, definitely interested. That's
okay, great.
Yep. So we have the version 1 of the handbook where we describe all these things will be available very soon. And that's meant to be version 1 to start the discussions. And then we can work on it and we'll have a new version every year.
Okay, yeah, great. Thanks.
Good.
Okay. Anybody else?
If not,
no more questions.
Well, it's
Friday evening.
It's Friday evening. It's 5:30.
Zoom fatigue.
Yes.
Hi, I'm Federico.
Hi, Federico.
Yep.
Hi. I was wondering if there's going to be a follow-up on this. If there's, well, Stefan just mentioned there will be some sort of a handbook, and how is this going to go on for all the stations and all the sampling that it's happening soon, etc.
So for the All Atlantic Ocean Sampling Day in 2022, the handbook will be fully developed so that we can it can be used for that event. More sampling that will happen sooner. You probably mean Mission Microbiome. Yeah. That will be, so we will have a separate set of protocols developed for that are based on that handbook.
Good.
Yes, we will meet anyways very soon about that.
Yeah.
Yes.
And maybe a question for Pierluigi, and an interesting presentation, complex topic, I would say. I was trying to understand if there was any chance to use also to start inputting past data into all these observation, or if it's only happening like from now that we will develop an ocean observation system. Thank you.
So on the data front, so on the ocean best practices front, the methodology, for sure, legacy methods are very interesting too, because it gives you a perspective on how the older data was collected and if it's compatible, how to analyze it. So that for sure can be added. In fact, people are finding methods that they've had on a floppy disk somewhere in a drawer, and they're like, great, somebody cares. And so they put it out there. In terms of the data, yes, that's also, I mean, many systems receive data that's very old. There's even that Biodiversity Heritage Library initiative, which is scanning old herbaria and written documents from the 1700s and digitizing them and putting them on maps again so we can track things. So old data is of high interest. That's how we know how the world has changed. But along with that data should come the methods, if possible. Should come together if possible, because then we understand how to treat that data. So the short answer is yes. Sometimes it's a bit more complicated. Please talk to the aggregators, because they usually have some kind of flow in place to help you do that.
I see that Kelly wants to say something.
Just I was on Stefan.
Offline, let's work out. There's some Mike Weiss is here from ONR. Let's get together to bring the Ghost Ship and the NOAA. It's time to have another meeting offline and get that going, seeing what we can do to get us to the next level, other than just, hey, so-and-so talk to so-and-so, but get some real plans going.
Yes.
I know our calendars are like crazy. And everyone come to the All Atlantic Side event. Can we advertise that real quick before you go?
If we have a screenshot, we can.
You know, but like, can I very quickly pick up on what Kelly just said? So this, like, you know, we talk to each other a lot, and it's hard to get stuff done because we try to do it between our projects. But in the next round, in the next set of EU calls or global calls, let's try to write it in that we're going to collaborate cross projects, you know, to make these things happen, to make sure that the protocols are synced and data is interoperable. Because we can still sort of compete in the typical academic sense, publishing papers, but at the same time engineer a system that's going to last in the long term. So I'd really encourage us to transform those short conversations into plans to, even if we're in different consortia, different grants, different calls, if we have similar data or similar methods, let's find a place to share them more effectively.
I'll share my screen. So this is the event that Kelly was talking about. It's a number of panels organized during the All Atlantic Forum, and it will happen on the 2nd of June at 16 100 UTC. And we'll have lots of 3 panel discussions about microbiomes and how we can push it to the next level.
And there will be pledges, and one of the pledges is the special issue that's coming up in Frontiers. But we could also, think about this group, thinking about the harmonization of methods as a pledge, right? So a lot of these pledges are stuff we have to do anyway, so we should think about adding that one as well.
Okay.
There's also another side event. So Frank mentioned just before the microbiome side event, there's an MBON side event. So the link is in the chat as well.
Thanks for that, Frank. We really do want to try to, encourage both of those as one contiguous side event. So we should try it. We got to talk more, Frank, so we can tweet each other's events. I know we spend so much time talking. It's like, and yet there's still so much more to talk about.
Okay, I don't think there are any more questions. There were just a couple of comments saying that something might be also needed for third-party cruises, something short and technical, from Daniele and Alessandro.
Okay. So I think it's probably time to wrap up. I'd like to thank Pierluigi, Anna, Eloise, and Colomban as well for joining us and presenting different parts of the webinar. And I want to thank all the participants for being there today and also for participating to the survey, which is very useful, as I said already. And we will be in touch for the next steps and probably with a survey that is more elaborate that will include other types of protocols as well.